metabolism of lipids and metabolism of saccharides. And today and tomorrow we will focus on the last group of nutrients that we utilize in our body, and that are amino acids. Today we will focus on the topics that describe the general possibilities what you can do with the amino acids. And before it, we will also mention something about the metabolism of proteins and metabolism of amino acids from the whole body point of view. Tomorrow we will look at the specific pathways that each amino acid can undergo, so how they can be utilized, how they can be synthesized, those that we can synthesize, and also what specific compounds we produce from amino acids. So if we start with the first part, I should give you some information about the general turnover of amino acids and proteins in our body. Maybe you can think that proteins are stable, but it's not fully true. There are many proteins that have very, very short half-life. Typically, that are the proteins with some signaling function. Typically, that are enzymes. Here you have, as example, 3 tissues: skeletal muscle, liver cells, and then the small intestine. And here you have the percentage of the daily protein turnover. That means that it's the amount of proteins that you degrade each day and that you subsequently resynthesize. So if we look at skeletal muscle proteins are quite stable. The daily turnover is only about 10%. If we go to the liver cells, there the turnover is much, much higher. It's around 40%. And quite simple explanation for it is that inside liver you have many enzymes that regulate our body metabolism. And during your day, your liver cells must change their metabolism in the period of starvation and in the period after your meal. That means that quite high amount of proteins must be destroyed in improper occasion, and on the other hand must be resynthesized when you return, for example, to the well-fed state. And the last example, that's the cells in small intestine. That we mean the enterocytes, that means the epithelial cells in the small intestine. And here the turnover is very, very high. The global turnover is around 80%. What's the half-life of enterocyte? So what's the typical life or length of the day?
120 days.
No, that's for erythrocytes. No, for enterocytes. That means the lining of small intestine. It's around 3 to 5 days. That means that they have very intensive metabolism. They are in close contact with not only useful compounds from our diet, but also with many harmful compounds that we have in the diet. So therefore, they must have very intensive metabolism. They must produce transport proteins, produce enzymes, and they do it really, really quickly. So 80% of the proteins are synthesized and degraded each day. If we take a global sum of the amount of proteins that we have in the standard diet each day, it's around 100 grams. Yes, that's the average number. And since we do not have ability to store proteins, the same amount, typically if you are adult, you burn. That means that the oxidation is also around 100 grams per day. And if you take, or if you convert it to the amount of energy that your body utilizes, it's about 10 to 20% of energy. That means that proteins as the nutrients are at the third position from the point of energy. First are saccharides, around 50–60%. On the second position we have lipids, that's around 15–25%. And then on the last position we have proteins. But what's important is that inside proteins we have many essential amino acids that we need for our body. So they are not in the diet to give us energy only, but they are in the diet also to give us essential compounds. Here we have the simple balance of amino acid metabolism in our body. So here we have the proteins in the diet. Of course, the day can vary. You can have a day where you have lower amount of proteins, other day you can have higher. So here is some average that forms one source of the amino acid pool. Then the second source of the amino acid pool is the degradation of proteins that you have in your body. That means proteolysis. And each day you degrade around 300 to 500 grams of proteins. That's in the time when you have no special needs. That means each day some amount of proteins will be degraded, and it's around 300 to 500 grams. And the last source for this amino acid pools are amino acids that you synthesize in your body. That means the synthesis of non-essential amino acids. We have three utilizers of this amino acid pool. The first is the protein synthesis. That means that if you are adult, you have typically normal nitrogen balance. It means that the amount of proteins that you degrade is equal to the amount of proteins that you resynthesize. So it's around 300 to 500 grams of proteins. Of course, it can be different proteins, but the total sum is the same. Second possibility is that you convert amino acids to some derivatives. Here we have mentioned only some of them. So for example, amino acid glycine is used for the synthesis of heme. Glycine is also used for the synthesis of purines. And glutamate gives nitrogen to the synthesis of nucleic bases in general. And as you will see tomorrow, we have many other molecules, for example neurotransmitters, that are other derivatives of amino acids and must be each day produced from this amino acid pool. And the last portion, we can say that's the rest. That's the oxidation of amino acids. And as you know, from the whole body perspective, we can say that in amino acid we have two parts of the molecule. The amino group, or altogether the nitrogen inside, and the carbon skeleton. Amino group can be converted to ammonia or to urea. Predominantly in our body we form urea. It's around 95% of the amino nitrogen that goes to the urea, and normally only around 5% goes to the ammonia that you excrete in the urine. And with the carbon skeleton, you can do different things. It depends on the amino acid that you degraded, but as a simplification, we can say that you can burn it. That means carbon dioxide and water, or you can convert it to glucose, to saccharides, or to triacylglycerols, or you can convert it to ketone bodies. And about this we will speak tomorrow. About this we will speak today. So is this clear to you? The general numbers of proteins that we degrade, resynthesize, synthesize, and oxidize each day. Here is one more. It's written that we have some sources, then it's the pool, and there are the ways for utilization. As you know, we have no possibility to store the proteins. Therefore, if we are adult, we can define something that's a nitrogen balance. And if you are adult, you have this nitrogen balance in equilibrium. That means that the intake of nitrogen in your food, that means predominantly the intake in the proteins, is equal to the losses of nitrogen typically in your urine. For there you can simply measure it. Of course, the nitrogen you also lose, for example, in your stool. You can lose some nitrogen in your sweat. You lose it if your cells desquamate, that means keratinocytes from your skin. If you lose them, you lose, of course, some nitrogen. But the basic definition is the amount of proteins or the nitrogen in the proteins in the diet, and then the losses inside urine. For this, you can quite simply measure. This means that we are in equilibrium, that if one day you, for example, eat 2 times more proteins that you want, then you do not store them in your body, but you simply oxidize them. And the carbon skeleton you can convert to some other compounds, typically to triacylglycerols. That means that the nitrogen will be metabolized and you will lose it. The carbon skeleton you can store in the body, but since we call this the nitrogen balance, we do not count the carbon skeleton, we count only the nitrogen inside. There can be times in your life when you have the positive nitrogen balance. That means that the intake of proteins is higher than the losses, and therefore you must store some nitrogen in your body. Typically, that you produce some new proteins that you use as a physiological thing, for example, for the growth of your body. So positive nitrogen balance is typical for the periods of physiological growth. That means if you are a newborn, you have 3.5 kilograms, for example, and then if you are 20, you have 70 kilograms. And not small portion of these are the new proteins that you must synthesize. So the growth of individual is accompanied with a positive nitrogen balance. Other possibility when it's physiological to have positive nitrogen balance is if you have recovery from some severe illness. So if you, for example, have some trauma, broken legs, for example, or if you have some severe infection or some other severe stimulus, that in the period of recovery, you typically develop the positive nitrogen balance, since during the stress, you typically have the negative nitrogen balance. That means that during the illness, you lose the nitrogen, and then in the recovery, you have the positive, so you store the nitrogen in your body. And the last possibility is the non-physiological function, and that's when you actively administer anabolic hormones to the patient. Could you name me some protein anabolic hormones that you know? Androgens, that mean testosterone, or the derivatives that are used in doping in sport. Some other? Estrogens are weaker, but to some extent also. Or growth hormone and its derivatives, or insulin. Yes, the most widely misused are especially the derivatives of androgens, yes, in the doping. But if you give the other protein anabolic hormones to the patient, you will also induce the protein anabolic state that will be accompanied by the positive nitrogen balance. And the negative nitrogen balance is the opposite. So typical example of this is starvation. You have no amino acids in your food, no meal. Therefore, you must degrade your own body proteins to form new glucose molecules in gluconeogenesis, and you lose the nitrogen in the form of urea. Second possibility is if you have some severe illness, some trauma, your body metabolizes the proteins to give enough energy and enough substrates for coping with the disease and for the reparation of the damaged tissues and to treat the illness. And third, unphysiological situation that's much more common than the administration of anabolic hormone is the administration of catabolic hormones. What do you mean, or what do you think is the most common protein catabolic hormone that you administer to the patient? Glucagon you give in very, very little occasion. only as the first treatment of hypoglycemia. Cortisol, yes, or derivatives of cortisol, that means the glucocorticoids. Glucocorticoids you use for many, many diseases. Altogether, it's one of possibility in treatment of autoimmune disorders. For example, for treatment of Crohn's disease, for treatment of multiple sclerosis, or many kinds of skin diseases. There you use the glucocorticoids, and it depends whether you use only local glucocorticoids that have not so big systemic effect, or if you give the whole body glucocorticoids. If you give the glucocorticoids to the whole body, they will induce the protein catabolism in the body. And it doesn't matter if you have nutrients in the diet or not, you will simply cause the proteolysis typically in your skeletal muscle. And since here we still speak about the nitrogen and we altogether speak about the proteins, here is something that we can call the conversion rate. So approximately you can say that 1 gram of nitrogen comes from 6.25 gram of proteins. Of course, this is only average, but if you will read for the patient that the amount of nitrogen in the urine is this, so many grams, you can simply convert it, how big is approximately the loss of the proteins. Yeah, so if you, for example, in urine has 20 grams of nitrogen, that gives you the lab, you can calculate that it's around 120 grams of proteins that are oxidized. That's something more than it. Any question to this? To the nitrogen balance? So the last part is briefly to say how this whole thing is regulated. In molecular biology, you have spoken about the synthetic part. So the protein synthesis, that means transcription, translation, and post-translational modifications. Yes, this process you should know in detail, but we will not repeat it today, and also it won't be part inside the test. What right now we will slightly cover, and I think that you also heard it, is the way how our body is able to mark the proteins for the degradation. Here you see that different proteins have really different half-life. On the first, you have the enzyme. Here it's ornithine decarboxylase. It's only an example of some enzyme. You can read the values for other. But if we look for ornithine decarboxylase, the half-life is 11 minutes. That means that in 11 minutes you will degrade 50% of the concentration, and if you want to continue in the reaction, you must resynthesize it. So the half-life is very, very quick. Second, here you have hemoglobin. Hemoglobin, as you know, is produced in the precursors of erythrocytes. Mature erythrocytes cannot produce it, and also they do not degrade it. That means that the hemoglobin molecule must stay in the erythrocyte for the whole life of the erythrocyte, and that's around 120 days. So here you see that the protein that must have the half-life much longer. And then here, the last, you have one extreme example of protein. That's the gamma crystalline. That's the protein of our islands. And island is produced during the development, but once it's produced, the cells that are able to produce it, they disappear. So you do not have them anymore. And therefore, this protein must stay with you for the rest of the life. So you can say that the half-life is 0. So it must stay, or not 0, but very, very long. For it must stay with you, for example, 60 years. And if you develop the problem in the lens, the only treatment is to remove the lens and put there a new lens, as we have no possibility to force any cells to repair the lens. Simply, there are no cells. And the good question is, how is this possible, as the different proteins have so different half-life? And the way how it's regulated is the ubiquitin and proteasome system. Have you heard about it? So, quick repetition. Ubiquitin is the marker, or it's the label for the protein to be metabolized. You know that there are 3 enzymes that are putting this label on the protein. The last one, the E3, means the ligase. We have around 600 types of this ligase that put the ubiquitin on the protein molecule. And different proteins have different affinity to these enzymes, and therefore different proteins will have different half-life. Those that have very big affinity to these enzymes, that means that they are eager to be ubiquitinated, they will be much quicker degraded in the proteasomes. Since the polyubiquitination is the marker or is the signal for degrading of the protein in the proteasome. The proteasome is the organelle that has the proteolytic activity. This whole process is highly regulated, and it explains us why different proteins have so different half-life, and also how our cell is able to regulate this whole process. What's the other possibility how to degrade proteins in the cell? Lysosomes. And what's the role of lysosomes if you compare them with proteasomes?
It's by pH. It has a low pH.
That's true. They have low pH, and they typically eat the waste. That means the bad organelles or the proteins from outside the cell. Yes. But for these enzymes and many of these signaling proteins, you use the ubiquitin proteasome system. Yes, not the lysosome system directly. And this discovery was so important that in year 2004 there was the Nobel Prize in Chemistry given by the discovery of the ubiquitin-mediated protein degradation. Who is it? Nothing? Good. So it's really the fact that if we will ask you what you can regulate, typically the students say the transcription. Yes, transcription we can regulate, we have the transcription factors. But it's not fully true. You can regulate also the lysis, and we have one big complex of many proteins that are able to do it. But we simply say that you do the proteolysis. But behind this, there are many steps that lead to the final decision, what's the half-life of the protein. So any question to this? So if no questions, then before we come to the general pathways, we will repeat those 20 amino acids that we should speak today about, and tomorrow also. As you know, we have quite a big number of amino acids. But those 20 that we have here, or 21, we are encoded in the DNA to be incorporated inside the proteins. We can divide them into 3 subgroups. The first subgroup are those that we can simply synthesize in our body. That means that we have the pathways to produce them, and therefore we call them non-essential amino acids. Simply, it doesn't matter if you have them in the diet, you can simply produce them. For example, glutamate or glutamine you can produce from alpha-ketoglutarate that you have in the Krebs cycle. Alanine can be produced from pyruvate that you simply gain from saccharides. Then we have, and these are around 10. Then we have those that we cannot synthesize in the body. And as you know, the compounds that we cannot synthesize, we call essential. Typical example are vitamins, since with majority of vitamins we cannot produce. For example, vitamin C. And these 8 amino acids we also cannot produce in our body. If we look what they are all branch chain amino acids, that mean valine, leucine, and isoleucine. 2 aromatic amino acids, that's phenylalanine and tryptophan. Tyrosine is missing since phenylalanine can be simply converted to tyrosine by 1-hydroxylation. That means if you have phenylalanine in your diet, then tyrosine is not essential to you. And then you have one from each group, one from basic, one from sulfur-containing, and one from hydroxy-containing group. From basic, you have lysine. From sulfur, you have methionine. And from hydroxy group containing, you have threonine. And then between these two groups, you have something that we call conditionally essential. That means that we have pathways to produce them, but there are some situations in your life when these pathways are not able to cover your demands. Typically, that are the period of your growth. You know that in childhood you have 2 big growing spurts, and in these moments you need really higher amount of proteins, higher amount of amino acids. Or if you have some severe illness or recovery from severe illness, your own synthetic pathways are able to produce some amount, but not all or the whole amount that you need. And therefore we call them conditionally essential. And here we have arginine and histidine. If you open your books, you sometimes can read slightly different distribution. Yes? Sometimes these 2 amino acids are in non-essential group. For the periods when they are essential is very, very long, and simply we have pathways how to produce it. Or you can have the arginine and histidine here in the essential group, since there are some periods when they are really essential for you. So if you will follow this distribution, you should be able to answer the statements in the test, but sometimes in the book you can read slightly different distribution. Yes, and the biggest difference is typically whether you have 3 groups or only 2 groups, and where are these 2 amino acids. So any question to this? So if not, then we will start with the metabolism of the amino acids. As you know, the amino acid is defined by the presence of 2 functional groups in the molecule, carboxy group and amino group. Then you have their hydrogen, and then you have here the side chain. For a glycine, is the side chain very small, and it's not chain, it's only hydrogen. But then for the majority of amino acids, you have here some carbon, more or less complex side chain. What we can do with these functional groups? First possibility, what you can do is to remove this carboxy group. If you remove it, you remove carbon dioxide, and you produce from it biogenic amines. Why it's called amine? Since that's the second functional group that you keep in the molecule. Biogenic amines are very important molecules. For example, all catecholamines are biogenic amines. That means adrenaline, noradrenaline, and dopamine. Quite big number of neurotransmitters are biogenic amines. Dopamine is neurotransmitter, noradrenaline is neurotransmitter, and also, for example, serotonin is biogenic amine. Only you take different amino acid for the decarboxylation process. Second possibility, what you can do is to modify this amino group. So if you modify this amino group by deamination, you remove ammonia from it. We call this oxidative deamination, and as the result, you will produce alpha keto acid. So by deamination, you convert the amino group to keto group, and from alpha amino acid, you produce alpha keto acid. Typical example, here you have glutamate, and here you have alpha ketoglutarate. And you remove ammonia, plus you oxidize the whole molecule. And third possibility, what you can do is that you take a pair of amino acid and one alpha keto acid, and you let them exchange their functional groups. That means that at the beginning you have the amino group on the amino acid, and after the transamination reaction you will have the amino group on the second acid. And the alpha keto group will be given to the amino acid. That means that you do very similar result as by deamination, but for transamination you need the pair, amino acid and alpha keto acid. So that means that here, other one. They only exchange the functional groups. So here you remove no ammonia from the molecule, you only exchange the functional groups between alpha keto acid and amino acid. Last possibility, what you can do with amino acid is the formation of peptide bond. That means that you will utilize this, and you will join the amino acids together. But about this last part, you spoke previously with biologists, so we will today speak only about these reactions, about the transamination, about the deamination reaction, and about the decarboxylation. You oxidize it during the reaction, and you gain from it typically the NADH. We will go right now through all these reactions. Only here you have the overview what kind of reactions you can do with these two functional groups. So, first is transamination. The transamination you have on this picture. So, here we started with the amino acid, for example, glutamate. You take some alpha keto acid, and as you see in the whole reaction, you simply change these two functional groups. So as the result of the transamination, you have alpha ketoglutarate and alpha amino acid. So the amino group from glutamate was transported to the alpha keto acid, forming the alpha amino acid, and here we have the glutamate. This reaction is fully reversible. So you also can start with some alpha amino acid, let it react with alpha ketoglutarate, and you will gain glutamate and alpha keto acid. This reaction, or this type of reaction, is metabolized or catalyzed by enzymes that we call aminotransferases or transaminases. These enzymes are specific for one pair of amino acid and its corresponding alpha keto acid. That means that you have quite big list of transaminases in your body, enabling the transamination for variety of amino acids. What's common for all of them is that they must have some cofactor for their activity. And the cofactor is pyridoxal phosphate. That's the derivative of vitamin B6. That means derivative of pyridoxine. Yes? And pyridoxal phosphate is the active cofactor that's utilized in all these transaminations. You have many of them in your body, but what we want you to know are at least these two. Since these two, that means alanine aminotransferase and aspartate aminotransferase, you will use quite routinely in your clinical practice. And you will meet them under the name of the liver enzymes. Do you know why do we call them liver enzymes? And why not, for example, pancreatic enzymes or brain enzymes? Inside the liver you have many enzymes, yes, but why these we call liver enzymes? Here. They can happen elsewhere. That's true, both of them we can use in gluconeogenesis, but it proceeds also in kidneys. The fact is that these two enzymes, or elevation of their values, very well correlate with the damage of the liver. So you typically use them as the marker of liver damage, and their values quite well correspond with the level of the liver damage. So ALT and AST are two transaminases that you use as the screening. For example, if the patient comes to you, to the hospital, and you do some basic list of examination, and you want to see the basic nutrients, basic ions, and then basic organ function, or better to say, organ damage. And ALT and AST are routinely inside.
And like, how do you check if they are in high concentration? If it's damage or
yes. Better to say than concentration is activity. Since, as you know, for enzymes we measure the activity. So if the activity of ALT and AST in liver, in blood plasma is increased, it's suspicion on the fact that there is the liver damage. And if you, for example, have infectious hepatitis or some acute liver toxin, you can have 20 times higher activities than normal of these two enzymes in the blood. Here we have the reactions that they catalyze, and you should be able to draw them or to identify the compounds. So ALT, alanine aminotransferase, takes the alanine plus alpha-ketoglutarate, and the result is pyruvate and glutamate. So you transfer the amino group from alanine to alpha-ketoglutarate. Or you can take it from the opposite. That means pyruvate plus glutamate, giving you alanine and alpha-ketoglutarate. AST, aspartate aminotransferase, takes aspartate plus alpha-ketoglutarate, giving you glutamate plus oxalacetate. Or you can do it in the reverse direction. That means that the aspartate tells you what, or the first tells you what you will transaminate, aspartate or alanine. As you see, the transaminases typically work with alpha-ketoglutarate. Yes, they work with alpha-ketoglutarate, or some of them work with oxalacetate. But these two alpha-keto acids, they are the predominant pair for the amino acid to be transaminated. Where do you think these enzymes will be located inside the cell? AST and ALT. They have different compartmentalization. So one will be in cytosol and the other will be... So ALT in cytosol, who is voting? AST cytosol, AST mitochondria, and ALT mitochondria. So the majority tells the right thing. Why ALT in mitochondria? Sorry, why ALT in cytosol?
Because pyruvate made in glycolysis in cytosol.
Yes, pyruvate you have in the glycolysis. Yes, it's not an explanation, but it can help you. And oxalacetate you have in Krebs cycle, and that's in mitochondria. And other explanation will be that this production of aspartate you will use in urea cycle also. But you can remember oxalacetate in Krebs cycle, that's in mitochondria. Alanine, it's pyruvate, that means cytosol glycolysis. This distribution is important for you. For if you have only elevation of ALT, that means that the hepatocytes are damaged, but you can say that only their cell membrane is damaged. And the mitochondria are intact if you have no big elevation of AST. But if you have, in the patient with liver problems, elevation of both ALT and AST, you know that there is some severe damage for both the mitochondrial membrane and the membrane of the cell. Both are damaged. And therefore, the intracellular enzymes can go to the bloodstream. So therefore, you should know the distribution, for it to some extent correlates with the severity of the liver damage. ALT is in cytosol. That means if you damage cell membrane, it's released. But AST is in mitochondria. So if you damage only cell membrane, it will stay in the mitochondria. If you damage both mitochondrial membrane and cell membrane, both are inside. So any question to this? Second is conversion glutamine and glutamate. We have raw alpha-ketoglutarate, glutamate, and glutamine. Glutamine is for our body very important molecule. The problem is that ammonia itself is for our body toxic. Yes, I will later, in the second part of the lecture, tell to you why ammonia is toxic. Right now, take an information that ammonia is for our body toxic. And therefore, our cells want to prevent the increase of ammonia in your blood. And one way how to prevent this is incorporate ammonia to some molecule. And very elegant thing is to incorporate it to alpha-ketoglutarate and glutamate molecule, forming glutamine. Since in glutamine you have altogether, we can say, 2 ammonias, and the cost was only 1 alpha-ketoglutarate. And glutamine is the main transport form of ammonia, or you can say amino groups or nitrogen in your blood. And ammonia and glutamine has the highest concentration, due to this fact, from all your amino acids in your blood. The concentration of glutamine is around 0.6 millimoles, and the concentration of ammonia is below 35 micromoles. So you can say that the difference is more than 20 times. Yes, 20 times more glutamine you have than the ammonia. If all molecules of glutamine lose the ammonias, you will reach the toxic levels of ammonia and you will be quite quickly dead. So glutamine is a very excellent way how to store the ammonia and transfer it through the body. If we look how our cells do the conversion of glutamate to glutamine, and then subsequently from glutamine to glutamate, you have it on this picture. So you take the glutamate and you take 1 ammonia molecule, and you will produce here the amide bond. That means that here the carboxy group is converted to amide, the ammonia is bound to it, and you must pay the energy, the ATP, to it. The enzyme is called glutamine synthetase. It's inside our cytosol, and this reaction typically proceeds in many tissues, in many peripheral tissues in our body. If you have ammonia in these tissues, they will incorporate it to glutamate, and they will send the glutamine that was produced to your blood, and this glutamine is transported to your liver. Inside your liver, you do the opposite reaction when you convert the glutamine to glutamate by enzyme called glutaminase. As you see, it's the simple hydrolysis, so you use water to destroy this bond, So you destroy the amine bond and you produce the carboxy group. Here you release the ammonia. And why do we do this typically in the liver? Since inside the liver we have the urea cycle where you detoxify ammonia by forming urea.
But what is the NAD plus under the glutamine?
That means that here you also do something with the oxidation. Yes, but right now you do not have to take into account.
Okay. Thank you.
So this is the elegant way how the toxic ammonia from peripheral tissues is transported to the liver mitochondria, where you release the ammonia, but you quickly detoxify it in the urea cycle. And therefore, by this transport mechanism, you can transport the toxic thing through your body without any toxic effect to you, and you will produce the ammonia directly in the liver mitochondria where you have the first reaction of the urea cycle. And therefore, this enzyme glutaminase is also in the mitochondria. First reaction of urea cycle in mitochondria, therefore the reactions producing ammonia also inside the mitochondria. So you should not think that these are the opposite processes, that here costs you ATP and here you produce nothing. It's the processes that do not proceed in the same compartment, in the same organ. Typically the glutaminase you have in the organs that can get rid of the ammonia. That's typically the liver tissues or the kidneys that can excrete the ammonia in the urine. And glutamine synthetase is in the peripheral tissues to consume the ammonia that's toxic for you. So do you understand this?
The glutaminase also exists in the peripheral tissue?
It can exist there, for it's typically in your brain, for example, for the glutamate recycling, but it's much more specific, yes? At this point of view, we can simplify that it's typically in the tissues that want to get rid of the ammonia from your body. That means the liver and in your kidneys, but you produce the glutamate cycle, for example, in your brain at the glutamate neurotransmission.
And like in the liver and the kidney, they exist like in both cases in the mitochondria?
Yes. Last, before the break, is how to convert glutamate to alpha-ketoglutarate. This enzyme is example of the oxidative deamination. That means you have amino group and you convert this amino group to keto group. And you release ammonia and you oxidize. So you gain the NADH or NADPH. This enzyme is able to give you both. This enzyme is mainly inside the liver, and the same as glutaminase is inside the mitochondria. Since the free ammonia can be consumed there by the urea cycle, where you detoxify it. So if we look at the reaction, we have the glutamate plus the glutamate dehydrogenase, and you oxidize it, plus you deaminate it. That means that you release the free ammonia. From glutamate, you produce alpha-ketoglutarate. The ammonia molecule enters the urea cycle. These cofactors can be reoxidized in the mitochondria. And the alpha-ketoglutarate can enter the Krebs cycle, or it can enter other transamination, for example, with ALT or with AST. So if we say the typical theory of the reaction is that you have amino acid with an amino group. This amino group you transfer to alpha-ketoglutarate, forming glutamate, and then you do the oxidative deamination of the glutamate, forming the free ammonia that enters the urea cycle. Not all amino acids do it in this way, but those that we mention here, for example, aspartate, alanine, they do it in this way. So do you understand this? So the ammonia is produced in two reactions, basically, by the glutaminase and then by the glutamate dehydrogenase reactions. And subsequently, you detoxify it in the urea cycle. That was set. So any questions? So if no questions, then we will take the break till 30. So in the second part, we will do the urea cycle. In the urea cycle, you simply want to prevent the accumulation of the toxic ammonia. If we draw a line of reactions, we can start with alpha ketoglutarate, converting to glutamate, and converting to glutamine. You know the reactions, so you know that glutamate to glutamine is glutamine synthetase, from glutamine to glutamate is glutaminase, and from alpha ketoglutarate to glutamate you have transamination, and opposite you have the oxidative deamination. What's the problem if the ammonia accumulates in your body is that you can say that in each of these reactions you add one ammonia. It means that if you have excess of ammonia in your body, it will shift the balance of these two reactions in favor of glutamate and subsequently in favor of glutamine. And this can have some severe consequences for your body. If you decrease the amount of alpha ketoglutarate in your cells, you took the intermediate, or you take the intermediate of Krebs cycle. That means the final place for the oxidation of each of all nutrients. And this, what we can call a block of Krebs cycle, will harmfully defect the production of ATP. And these cells will start to suffer from lack of ATP. If we then shift to the glutamate level, glutamate is very potent neurotransmitter. Glutamate is the most common excitatory neurotransmitter of our brain. Yes, around 40% of our synapses do the action through glutamate. Plus, it can be decarboxylated to other neurotransmitter that we call gamma-aminobutyric acid. And that's the most common inhibitory neurotransmitter of our brain. If you combine these two together, you have around 80 or maybe even more than 80% of all synapses. That means that if you artificially increase the amount of glutamate and GABA, by the fact that you take the alpha ketoglutarate and you let it convert to glutamate, you will cause the disbalance of neurotransmitter systems in your brain. And one of the problems in the accumulation of ammonia is the disbalance of the neurotransmitter systems. And in further years, you will heard about the state of liver encephalopathy. That means the harmful effect of your ammonia on your brain if it crosses the toxic concentrations. And the last is the glutamine. If the glutamine increases in your body, there is one big problem for your brain. Typically, your brain that has quite intensive metabolism of these neurotransmitters sends the glutamine to your blood. When the plasma concentration of glutamine rises, there is not so big possibility to push the glutamine into the blood, and you can say that these compounds accumulate. Plus, you must take into account the fact that the neurons are missing energy, since they do not have the alpha-ketoglutarate for the Krebs cycle, therefore they do not have ATP, and therefore they do not have energy for the pumps in the membrane. So the accumulation of the osmotically active particles, limitation of the export of these particles outside the brain, outside the CNS, plus the lack of energy for keeping the ion balance on the membrane will lead to the state of the brain edema. And as you know, the volume of the skull has some maximal amount. If you have the brain edema, then in some situation or at one moment, the brain will come to the intimate contact with the bones, and if you should judge the fight brain and the bone, you will know who will win. So the result will be the compression of the brain, and this at the very end can lead to the death of the patient. So right now I slightly describe the pathogenetic mechanisms that are behind the excess of ammonia, how it modifies your cells, and predominantly what you should know is why it's so toxic to our brain. Yes? In brain, you have very highly intensive energy metabolism. It consumes 20% of your energy. Therefore, if you limit its energy production, it's a very big problem for it. Plus, you put a disbalance into the neurotransmitter systems, those two most common neurotransmitter systems in the brain. And third, you limit the export of osmotically active particles outside the brain. And therefore, your body is really trying to keep the ammonia at a very, very low level. If you are adult, then below 35 microliters. In different periods of your life, you have different physiological values. Yes? If you look to the tables, you see that in newborns, you have slightly more ammonia that the newborns can withstand. Yes? But for adult, the value is around 35, 40, 45 maximal. And if you have higher values, for example, double times higher values is really toxic for you, and the first organ where you will see the dysfunction is typically your brain. And the patients that have liver cirrhosis can die due to this liver encephalopathy if you are not able to get rid of the ammonia from your body. So do you understand why ammonia is toxic? This is not the only reaction that's modified. The same you do with oxalacetate, aspartate, and asparagine. Yes, oxalacetate will be also taken from the cycle, from the Krebs cycle. But on the alpha-ketoglutarate, if you will be able to explain it in the test, in the exam, in the second year, it's absolutely enough for you. So if no question, then we will look how do we detoxify the ammonia. The urea cycle is nothing very difficult. The substrates for the urea cycle are all compounds that you already know. It's aspartate, that means one amino acid, that will bring one nitrogen into the synthesis. Then it's carbon dioxide, and the last substrate is ammonia, and from them you will produce urea. That's the cycle. The only complication in the urea cycle is that you will meet there two non-proteinogenic amino acids. That's ornithine and citrulline. But as I will try to show to you, you already know them since they are very similar to arginine. So if you know arginine, you have no problem in drawing ornithine and citrulline. This pathway in full is located in your liver. Some enzymes you can meet in other organs, but in one big complex that have some quantitative effect for the whole body, you meet these enzymes inside the liver only. The beginning of the urea cycle is located inside the mitochondria. That's the place where you start detoxifying this ammonia. And subsequently, the intermediates are transferred to the cytosol, where you do the other reactions. All this, or this whole reaction, is quite consuming energy for you. Altogether, you will use 4 ATP molecules for 1 urea production. That means that you really must feed the urea cycle by some exergonic process. Therefore, you also do it in liver mitochondria, since you know that inside liver mitochondria, you typically have a lot of energy, even during the starvation, since you metabolize there the fatty acids. In urea cycle, you do not have to know all enzymes, but what we want you to know, at least, is this one enzyme that catalyzes the first reaction, and it's also the main regulatory enzyme of the whole synthesis. So you should know carbamoyl phosphate synthetase, type 1. Why type 1? For you will meet the cytosolic carbamoyl phosphate synthetase, that means not in mitochondria, but in cytosol, in the synthesis of pyrimidines. So you will see 2 isoenzymes. Today we will use the mitochondrial one for the urea cycle, and next course you will use the cytosolic one for the synthesis of nucleic bases of the pyrimidines. So if we proceed to the first reaction, here you have the reaction in 3 steps, but what you should be able to draw is that you take carbon dioxide, here in form of bicarbonate, plus ammonia that you take from the glutaminase or glutamate dehydrogenase, and you combine it together with the ATP, with the phosphate from the ATP, and you produce this molecule of carbamoyl phosphate. So this is the part of the urea, and all other reactions will be to put here the second ammonia. So this is the ammonia that was taken here. This is the carbon oxygen taken from the bicarbonate, and here the phosphate is taken from the second ATP. As you see, the first ATP is used for joining the ammonia plus carbon dioxide together. And then the second ammonia is here to produce the anhydride bond between the carbamic acid plus the phosphate. And this whole compound is for you macroergic, and it can condensate in the subsequent reaction with other compound. So 1 ATP is used for joining bicarbonate plus ammonia together, and second ATP is used for forming the macroergic anhydride bond in the carbamoyl phosphate. This reaction is located inside the mitochondria. In the second reaction, you take the macroergic carbamoyl phosphate and you let it condensate with the amino acid ornithine. If you look at the ornithine, it's really highly similar to arginine. Ornithine has 5 carbon atoms, 1, 2, 3, 4, 5. Here is the amino group, and here you have other amino group. As you will see in a few moments, arginine only here has 1 carbon and 2 times nitrogen. So if you know arginine, you are able to draw the ornithine, yes? And really you do not have to memorize ornithine and citrulline, only take the arginine and look what's the difference between. And then you, in the test, you can draw arginine and only do not draw some small part of it. Ornithine, in this second reaction, combines together with this part of the carbamoyl phosphate. That means that you will release the inorganic phosphate, you destroy this anhydride bond, and the energy that you release is used for the condensation with this nitrogen. And as you see, here we have carbon, oxygen, nitrogen, coming from the carbamoyl, and this is the ornithine. And the whole molecule is now called citrulline. So citrulline has 6 carbon atoms. It has 3 nitrogens inside, and the difference from arginine is here. For here you should have the nitrogen. So do you understand it? How this reaction proceeds? This citrulline, you in next step transfer into the cytosol. So the place where you shift from mitochondria into the cytosol, that's the level of citrulline. Any question? No. We utilized ammonia, carbon dioxide, and now in the subsequent step we use the third substrate, and that was the aspartate. You want to incorporate this amino group into the molecule, and therefore, as you see, you will use the ammonia for joining the citrulline molecule. So it's other condensing reaction, therefore you must use ATP to feed this whole process with energy. So this is the third place in the synthesis where you use the ATP. And here you form the more complex molecule of the synthesis. Its name is arginosuccinate, and if you look at the structure, it's not difficult, since here you have the arginine. This is the amino acid arginine. And here you have the decarboxylic acid succinate. So one carboxy group, second carboxy group, 2 carbons in between, that means 4 carbon atoms inside. So this is the biggest conjugate that's named arginosuccinate, only describing the 2 components that are inside this molecule. And now what you do are 2 processes where you release something from this big complex. First, what you want is to remove the succinate out of the molecule. So the enzyme is called arginosuccinase. Simply, you remove one part of the molecule from it, therefore the ending ase to the substrate, and you destroy this bond, forming the arginine, and here you form the double bond. That means that you release the fumarate. So the electron from this bond will form the double bond. And then in the last step, you want here to destroy this bond, and here you can release the urea. So that's the second lytic reaction where during the hydrolysis of arginine, you destroy this bond. The oxygen you incorporate or you bound it to the carbon, and here you gain the molecule of urea, and you regenerate the molecule of ornithine. Now the ornithine can return inside the matrix of mitochondria. It can go to the second reaction with carbamoyl phosphate, forming citrulline, then joining aspartate, and then you cleave it to other molecule of urea. So as you see here on this colorful image, you see that the sources of ammonia and water are different. One ammonia comes from aspartate, or one nitrogen we can say comes from aspartate. The other comes from the ammonia itself. Carbon comes from the carbon dioxide molecule, and then the oxygen comes from the water. Yes? You do not have to know it in so big detail. What we want you to know is that one is from ammonia, nitrogen, and the other is from aspartate. And you saw here the whole enzyme process or catalytic process behind. So any question to this? Good. If you have no question, then we will look how this pathway is connected to Krebs cycle. You saw that one of the intermediate of the urea cycle is aspartate, and one product of the urea cycle is fumarate. And the way how you regenerate fumarate to aspartate is by using some enzymes of the urea cycle. Of the Krebs cycle. So where do you produce the aspartate? That's quite simple. It's from oxalacetate, and the enzyme is called AST, aspartate aminotransferase. So you take oxalacetate plus glutamate, and the glutamate can give one of the amino group to the oxalacetate forming aspartate, and here you have the alpha ketoglutarate. Then the aspartate joins the citrulline molecule, forming arginosuccinate. And in the next reaction, where you release arginine, you also release fumarate. And this fumarate you modify to oxalacetate by two enzymes of the Krebs cycle. In first reaction, you add water on fumarate, forming malate, and then by malate dehydrogenase, you produce oxalacetate. And then in mitochondria by AST, you convert oxalacetate to aspartate. So if Krebs cycle is not working for you, the urea cycle cannot work also. One reason is this, that means that it regenerates the fumarate to aspartate. Second is that for urea cycle you need ATP, and Krebs cycle, that's the final place of oxidation of all nutrients. If you do not have Krebs cycle, you do not regenerate aspartate and you do not have energy. Therefore, urea cycle also cannot work. Any question to this?
The NH4 comes from the glutamate, from the glutamine for this reaction?
You can say that it comes from both. It depends what you have in excess. So it can come from glutamine by glutaminase, or from glutamate to alpha ketoglutarate by glutamate dehydrogenase. You can do it. Or you can, from glutamine, go to glutamate, you can say, by glutaminase, and then the glutamate is transaminated with oxalacetate, giving you aspartate. Yes, there it depends what is the real situation inside the cell, whether you have a lot of glutamine or not so many glutamine and some level of glutamate. That depends on the situation. But all these three processes are plausible, can happen. Of course, ammonia is produced not only from amino acids. Yes, it can come also from other molecules. So this ammonia sometimes is not from amino acids, and then from glutamate you do only this transamination. Typically, for example, your bacteria in your intestine produce the ammonia, and your liver cells must detoxify this ammonia also. Or your enterocytes, to some extent, must detoxify. So the last is the regulation. The regulation can be divided to three parts. First, what we can say is only describing part is that, as you know, urea cycle is last part of the utilizing of proteins of amino acids. That means that if you will have high protein diet, that means that you or your colleague will eat high amount of proteins. It means that he will have high level of amino acids. As you know, they cannot be stored in the body. They must be oxidized, and therefore the ammonia must be detoxified in urea. Therefore, if you have high protein diet, you typically can see the increase of the transcription of the enzymes of the urea cycle. Simply, you must detoxify the ammonia. Second reaction, or second regulation, is regulation related to acid-base balance and intracellular pH. In urea cycle, if we do a simplification, you can say that you convert base called ammonia to neutral compound called urea. That means that from your body you remove base and produce neutral compound. The question is what you will do with the H+, that means hydrogen cations, that are typically bound to this base. Answer is simple, they will stay in the solution. That means that from the cell level, urea cycle is proton productive reaction. You do not produce there any acid, but you consume base. And for the body, the consuming of base is the same as production of acid. You simply keep hydrogen protons in the environment. And therefore, the urea synthesis must be inhibited if you have acidosis, that means low body pH. That's the situation where you will utilize the second possibility how to get rid of ammonia. If you have acidosis in your body, then your kidney cells will take the glutamine from the blood, cleaving it to ammonia, and this ammonia molecule they will put into the urine, and you get rid of this base, but this base has on it the H+. And if you have a severe acidosis, then the ratio 95% for urea synthesis and 5% of free ammonia excretion will be shifted to 50-50. That means that your kidneys will start to excrete around 50% of the ammonium from your body, and therefore they will also excrete the H+ that's bound to that base. The third is the direct regulation on the carbamoyl phosphate synthetase. This enzyme is the first enzyme that consumes the energy, and therefore it's also logic it to be the regulatory place. This enzyme is activated by the allosteric modulator that's called N-acetylglutamate. That's the derivative of glutamate if you take glutamate plus acetyl coenzyme A, and the acetyl is bound through nitrogen to the glutamate. You do not have to remember in detail this name of the enzyme that synthesizes N-acetylglutamate, but what you should know is that arginine, one amino acid, is activating this N-acetylglutamate synthetase. That means that arginine indirectly through allosteric modulator activates the whole urea cycle. So not only in general the proteins and amino acids in diet, but altogether the arginine through the allosteric modulator also modulate the velocity